Ever wonder what spermatogenesis is? Well, it is the process that occurs in the semi-infernous tubules of the testes that produce male gametes. It begins at puberty and continues throughout life. Supporting spermatogenesis are Sertoli cells that provide nutrients and signals to the dividing cells. Every day, a healthy adult male makes about 400 million sperm. Let's take a look at how sperm is made. Outermost tubule cells, which are in direct contact with the epithelial basal lamina, are stem cells called spermatogonia. Spermatogenesis begins during puberty, and from then on, each meiotic division of the spermatogonium results in two distinctive daughter cells, types A and B. The type A daughter cells remains at the basal lamina to maintain the pool of dividing germ cells. The type B daughter cells moves towards the lumen, where it becomes the primary spermatocyte, where it is destined to become 4 sperm.
Before you can create sperm, the cell must undergo meiosis. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are sex cells, sperm in males, eggs in females. Meiosis can be split into several stages between the first time the cell divides, meiosis 1, and the second time it divides, meiosis 2. The first cell division is meiosis 1, and it begins with prophase 1. In prophase 1, each chromosome is composed of 2 sister chromatids containing identical genetic information. The pairs of chromosomes may then exchange bits of DNA in a process called recombination, or crossing over. At the end of prophase 1, the membrane around the nucleus of the cell dissolves away, releasing the chromosomes. The meiotic spindle, consisting of microtubules and other proteins, extends across the cells between the centrioles. In metaphase 1, the chromosome pairs line up next to each other along the center of the cell, and the meiotic spindle fibers attach to one chromosome of each pair. In anaphase 1, pairs of chromosomes are pulled apart by the meiotic spindle, with one chromosome to each pole. Finally, in telophase 1, chromosomes move to opposite poles of the cell, membranes form around each set of chromosomes to create 2 nuclei, and the single cell pinches in the middle to form 2 daughter cells.
Transitioning to meiosis 2, prophase 2 contains 2 daughter cells, each with 23 chromosomes. The membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. In addition, the centrioles duplicate and the meiotic spindle forms again. In metaphase 2, the chromosomes line up end to end along the equator of the cell. In addition, meiotic spindle fibers at each end of the cell attach to each of the sister chromatids. In anaphase 2, the sister chromatids are then pulled to opposite poles due to the action of the meiotic spindle. This is where separated chromatids are now individual chromosomes. In telophase 2, the chromosomes complete their move to the opposite poles of the cell. A membrane forms around each set of chromosomes to create 2 new cell nuclei. In the end, there are 4 granddaughter cells, each with a half set of chromosomes.
To conclude spermatogenesis and meiosis is spermiogenesis, the final transformation of spermatids to sperm. In spermiogenesis, each individual spermatid elongates, sheds excess cytoplasm, and forms a tail. The head of the sperm, the acrosome, enables the sperm to penetrate and enter the egg. The middle section of the sperm, the midpiece, contains mitochondria. The final piece of the sperm, the tail, propels the sperm along its way in the female reproductive tract.